Assalamu alaikum. Welcome again to the next part of apoptosis, the extrinsic pathway of apoptosis. In the last video, we discussed the intrinsic pathway of apoptosis. If you want to watch that video, the link is in the description box. You can click there to watch that video. Now, why this pathway is called extrinsic pathway? Because it actually originates from outside the cell. The signals for death comes from outside the cell. That this cell must have to be apoptosed. So the signal for apoptosis comes from outside the cell, or it originates from outside the cell. That's why it is called extrinsic pathway. Now, what really happens in this extrinsic pathway? What happened that ultimately the cell will die or apoptosed? So now here this is a cell membrane or plasma membrane, and there is a receptor on this membrane, on this plasma membrane. We call it phase receptor. This receptor is called phase receptor. There are many receptors, as you know, there are many receptors on cell surfaces. Every receptor has its own function. So this is a specific receptor which actually play a role in apoptosis, which receive signals for apoptosis actually. And this phase receptor has some internal part. Inside cell, this receptor has some part, this part. This is called death domain. Why this part is called death domain? Because it actually binds or it activates other molecules inside cell which play a role in death, in cell death. So that's why it is called death domain. Now, what really happens when a signal comes, when a signal for apoptosis or for cell death comes? Here, this phase receptor, why it is called phase receptor? Because it binds to a molecule called phase ligand. That's why it is called phase receptor, because it receives signals from this phase ligand. And actually this phase ligand is present on the surface of T lymphocytes, cytotoxic T lymphocytes. So when cytotoxic T lymphocyte gets stimulated, that there are some cells, or there are cells which want to be apoptosed, or which must have to be apoptosed. So cytotoxic T cell comes here and binds to this cell through this receptor, through this phase receptor, this phase ligand which is actually present on surface of cytotoxic T cell. For example, this is cytotoxic T cell. So this phase ligand is present on its surface and it binds to this phase receptor. When this phase ligand binds to this phase receptor, now what happened next? Here, this death domain, there are some conformational changes occurs in this death domain. This death domain changes its structure, and when it gets activated or it changes its structure, what happened next? Here are inside cell some molecules called adapter molecules, which actually take signals from this death domain and activate the next pathway. So this death domain then binds or activates molecules called adapters. Adapters actually are a bridge between this receptor and other pathway inside cell which occurs for apoptosis. So these adapters bind to this death domain, or it actually activated these adapters. For example, there is adapter called FAD. FAD is for phase associated death domain. So it is actually phase associated. That's why these are called phase associated death domain. So when these adapters gets activated, These combinedly, this domain and these adapters are combinedly called disk. Disk is for death-induced signaling cascade. Because it induced death, that's why it is called death-induced signaling cascade. These all adapters in this death domain are combinedly called disk. So then next, what happens when these two combines? This disk activates some inactive caspases, which are called procaspase 8. This is an inactive pump. Procaspase is actually an inactive pump. When it gets activated by this disk, due to this disk, this procaspase 8 gets activated and it becomes caspase 8. When it's an inactive pump, it is called procaspase. When it gets activated, this is called caspase 8. So now this caspase 8 will activate further caspases. This caspase will actually activate caspase 3 next. So then what happened when caspase 3 gets activated? Actually, this caspase 3 is activated also from an inactive pump. This caspase 3 is also called procaspase 3. But when it gets activated, it becomes caspase 3. And caspase 3, as we already talked in the intrinsic pathway, that caspases are molecules, are enzymes, which must have to be activated for apoptosis to occur. So these ultimately, for apoptosis, these caspases must be activated. So when procaspase 3 become caspase 3, this caspase 8 activates procaspase into caspase 3, and ultimately apoptosis occurs. This caspase 3 goes and activates some enzymes inside cell. As we already talked about this, that there are some enzymes called proteases and nucleases, which are activated by this caspase 3, and proteases will go and cleaves, or it will degrade cytoskeleton inside cell. And other DNAs are endonuclease enzymes, which are activated by this caspase 3. This caspase 3 activates actually these enzymes, protease, and other one is endonuclease. Protease will degrade proteins. It will break or it will cleave proteins. And as we know that cytoskeleton inside cell is made up of proteins. So this protease will degrade cytoskeleton, and the cell and the cell structure will distort, cell shape will distort. So cell will ultimately gets into apoptosis. And endonuclease will go and it will degrade DNA. So if DNA has been degraded, so actually then apoptosis occurs. And here also there is also a link with intrinsic pathway. Here when this caspase 8 comes, here is a link with actually intrinsic pathway. What happens? This caspase 8 can also, this caspase 8 has the ability to cleave a molecule called BID. We were already discussed that BACs and BAC are molecules, BCL, which are related to BCL4 family. So this BID is also includes in BCL4 family. This is a member of BCL4 family. This caspase 8 cleaves BID. When it cleaves BID, BID will convert into T-BID. BID is a molecule. It will convert into T-BID. It means this is the active form of this BID. T-bit is the active palm of bed. So when T-bit produces, when T-bit is when bit is activated, this T-bit activates these two molecules, Bakes and Bake. And we were discussed an intrinsic pathway that Bakes and Bake produces here pores in mitochondrial membrane. So this Bakes and Bake produce pores in mitochondrial membrane. And some molecules like cytochrome C comes out from mitochondria. And cytochrome C then and cytochrome C with other molecules, with other pro-apoptotic molecules activate this caspase 9. When caspase 9 gets activated, so what happened? That here caspase 9 will activate caspase 3. So these both intrinsic and extrinsic pathway will combinedly undergoes both pathways.